Okay. Uh, learn something new. Okay, you're seeing my screen, right? We've seen the cursor move up and down.
Yes. Yeah,
Okay,
I see it.
that's all we need. Okay. Uh, what I was going to say before I shut it off for a second is that if I start the recording earlier, right at the beginning when we begin, okay, some of you are going to be filmed. Okay, you may have noticed that if you looked at the recordings. And I mean, in full color, full size, whatever. If you don't want that, if you don't want to be filmed at all, then you need to kill your video at the start because we will be starting the recording real close to the start. Okay, and that's a shame because it's nice for me to have faces to look at rather than blackness, darkness, uh, all all the time. Okay, here's our lecture topic today. It's not long. It's cellular processes and refers to the cell membrane and what it does and doesn't do uh, for the cell itself. Okay, the cellular process is any sequence of events. That's all it is. That always leads to something. It can be to chaos sometimes, but usually it's constructive. The processes are of the cell membrane and the cell intermembrane and the nuclear membrane. These are membrane related processes. These are divided into two categories. Passive processes. Passive processes, uh, these do not require work by the cell or organism in which they occur. How can anything go on and not require work? That's just doesn't make sense to most of us. Well, the work has already been done. The work has been done long ago in the formation of the universe, and we have molecules and atoms moving around already, each with their own inherent energy. Often we call that Brownian motion. Okay, but that's not anything that has to be developed now, today. So we call these passive processes. They don't require work. Some of these processes are going to seem very, very familiar to you, especially as I said, coming from Bio 20 and biology. The first one is so familiar, it's almost embarrassing to talk about, but we'll get over it fast. It's diffusion. Diffusion. Okay. Everybody, I think, is familiar with real life examples of diffusion. It's motion from an area of higher concentration to a motion of lower concentration. Okay. Can we think of a real life example? Or I can give you one and then you can try to give me one. Okay. Take a fishbowl. Okay. That could represent a cell if you wish, but I'd take a rectangular fishbowl. I don't happen to like those. And I'm going to take some dye, water or vegetable dye. I'll make it blue. I like blue. And I'm going to, with an eyedropper, put dye into the fishbowl very slowly and gently. I'm not swirling it around. Is the dye going to spread out over the entire fishbowl immediately? That's the question for you.
No.
No, it's going to take time. How long do you think it will take on a fishbowl that measures, a foot across and 8 inches deep and 8 inches high? You get the idea. Uh, Shoebox. Okay. How long would it take that dye to spread out? Any guess is correct here.
30 minutes?
30 minutes.
Yeah.
Okay. I not hearing any other objections to that. I'll object. It's probably going to take days. It's probably going to take days. It takes a long, long time. And that means it keeps on going until it reaches what's called equilibrium. Equilibrium is where the concentration of the solute, that's the dye, and the solvent, that's the water, are equal. The water is equally concentrated everywhere, and the dye is equally concentrated with the rest of the dye everywhere. So diffusion continues on till that point is reached. Once once equilibrium is reached, do you think the dye and the water stop moving?
No?
Okay. No, they don't.
No.
They move. Some dye will move higher in the fishbowl. Other dye will move lower in the fishbowl, and water will move this way and that way, but they still maintain the equilibrium. Okay. One thing that's important is that diffusion does not involve crossing of membranes. Okay. I said a lot of this was going to be on membranes, but diffusion, once material is inside the cell, it's free to diffuse. Once it's outside the cell, it's free to diffuse. Here in these beakers, they're showing, you can call it dye, being dropped here into the water, and then it spreads out, and then it spreads out until equilibrium is reached. But no membrane is involved. When I was in my first science class in 8th grade, I had no trouble with diffusion either. Nobody did. Then the teacher said, we're going to talk about osmosis, and about ready to drop out. Osmosis is only a special type of diffusion, and it's vital to life. It's the diffusion through a semi-permeable membrane. If you read along with me here, it is the diffusion through a semi-permeable membrane, with some things being allowed to cross and others being prohibited from crossing in one or both directions. Well, a semi-permeable membrane. I think that's when I wanted to tune out in 8th grade because I didn't understand a semi-permeable membrane. The teacher didn't do a damn good job of telling me what it was. A semi-permeable membrane lets some things through it and some things not. We have lots of semi-permeable membranes in life. And I wish you'd used an example. For instance, I've got a window in this room. What can come through? Well, I'll answer that. Light can come through. Okay. Uh, can a bird come through? I hope not. Okay. Can a wasp come through? I hope not. Okay. Can sound come through? Yes. So some things can come through that glass membrane and some things cannot. And that's the same with the cell membrane of the plasmalemma. Some things can come through, some things cannot. Some things, once they get inside, can no longer get back out. An example of that is amino acids generally entering cells freely, but once inside, they may be used for the production of larger proteins and DNA and not allowed to exit again. If you can use your imagination for a second and envision going into a room, closing the door, there's a window with a screen, and the screen is intact. It's fine. The window is shut, however. The window is shut on the inside. The screen on the outside is good. The door is shut to the room. It's actually hermetically sealed. There's enough air in there for you. And on the table, you have two jars. And one jar, one jar you have a jar of perfume. Okay. And in a second jar, locked up, you have flies, house flies. Now, open both jars. Will the perfume vaporize and go into the room? Don't let it be the same person now. Come on. Will perfume evaporate out of the jar? Have you lived?
Sorry, my button wasn't working. Yeah, I can.
Okay, then. Uh, there's a lot of buttons not working, huh? Yours, you got fixed. It was a space bar?
No,
Oh,
I
the
just
little
my laptop's been acting up lately.
Oh, okay. Uh, yes, of course, the uh, perfume can uh, evaporate out. It would potentially evaporate out into the room until it's at equilibrium throughout the entire room. Now, what about the flies that you open the jar? You think they're all going to stay in there? No, no, they're not. Beat you to that one. Some of the flies will come out now, some will come out later. Eventually, since there's nothing to entice the flies, they'll be all over the room. Okay. Both of those are examples of diffusion. Okay. You may say to me, well, the fly's using energy. Okay. Yeah, well, that's for a reason here uh, to purposely move. But what I want to get is going over to the window and open the window, but leave that screen intact. Can the perfume now also go out the window?
Yes.
Yes, it can. Thank you. Can the flies get out the window?
Yes.
No, not the flies won't be able to get out the window because we've got a screen and the screen is intact. Okay, not damaged. So some things can get in and out, others can't move through the barrier, which in this case is the screen. Okay. So this is an example of osmosis. Okay, not cellular osmosis. This is contrived osmosis using using the perfume of the fly model. Okay, but it does have to do with these three terms. Okay, these are all to represent red blood cells. Let's take the one in the center first. It says it's isotonic. Isotonic means that the solutes, that's the material that is dissolved in a medium, such as water. It could be table salt, it could be saline solution, and the solute on the outside and the solute on the inside are the same concentration. That will mean that the medium or water outside and inside are also of the same concentration. Okay. You put something like a red blood cell in a plasma that circulates through the body. So long as the tonicity or the strength of the solute and solvent are the same on both sides, we don't really expect any net movement to happen. Net movement. Instead, we expect the red blood cells to stay nice, well formed in the absence of any disease, of course. Okay. And so in an isotonic solution, we don't have that happening. Let's take a hypotonic solution. Hypotonic. Hypo refers to under, to less. And a hypotonic solution has fewer solids outside. You can consider it less salty outside and instead more water outside. The opposite is the case inside. There's less water inside. This is not in equilibrium, and this is going to move to equilibrium rapidly. You've got more water outside because you have less hypotonic, you have less salt outside. That greater amount of water outside is going to rush in so fast that the two sides don't have a chance to reach an equilibrium. And the water rushes in and blows up the red blood cells. Look how much bigger they are. And some are exploded. Now, if you have your book to this page or figure, I don't have a figure written on it. This is called hemolysis. Hemolysis. I'll find it for you. If you were to inject distilled water into a vein, it's the highest you can get, 100% water. It will flush into the area of red blood cells and explode them. Hypotonicity. It's important to maintain isotonicity. And then what's the first one? Hypertonic. Hypertonic refers to a very salty environment on the outside and less water. Water always moves to seek its own level. And so there's more water inside and it will rush outside. It will rush outside. That causes the deflation of the red blood cells. And this deflation is called crenation. This is used when people who have varicose veins behind the knee or wherever else they may have them, usually from sedentary jobs. If you have the blue veins, they will come and inject salt water. I'm sorry. Yes, salt water. And the salt water will cause the water in the red blood vessels to leave them and go into the vessel. It'll cause the red blood cells to crenate, to get smaller. And as you inject the salt water into a vein that is varicose, you can literally watch that vein disappear. Somebody might want all of their veins done at one time, but you would hurt your circulation. So you do some at a time if you have a large number. Now, this doesn't have a lot of verbiage up here to remind you what was said, but the verbiage comes below it. The isotonic solution has the same concentration of solids both inside and outside. Under these circumstances, there's no net movement of solvent, and in this case, the amount of water entering and exiting the cell remains the equal. So that's this one. Hypotonic is going to go to the last one, where you went. In a hypotonic solution, there's a higher concentration of solids inside the cell than out. When this occurs, more solvent will enter the cell than leave it. In this case, solvent is water. This balances out the concentration of solid. For an RBC, this results in hemolysis, or splitting or exploding. Hypertonic solution, this is the one, the opposite situation. There's more solute, more salt outside the cell than inside it. In this type of solution, more solvent will exit the cell, then will enter it in order to lower the concentration of solid outside the cell. For an RBC, this results in crenation or shrinking. When you are ever exposed to this concept again, and you will be, again in physiology, I'm sure, they're going to talk about the fact that water moves in or moves out, or in the case of the isotonic situation, stays the same. We'll talk about the motion of water, and that's absolutely true. But these terms, hypertonic, isotonic, and hypotonic, were developed first. And long before people were considering the movement of water, they were considering the movement of salt. Well, if one moves, the other moves as well. Okay. And so don't be confused, and don't let me confuse you right now. Hypertonic means that the solution that this cell is in is very salty. This is as salty outside as inside, and hypotonic is less salty outside. Facilitated diffusion. This is another type of passive diffusion. It's also sometimes called passive transport. Passive transport. When a molecule arrives at a cell membrane, whether from the inside or the outside, this shows the outside, it'll have a higher concentration outside, and it's going inside where it has a lower concentration. That meets some degree of the definition of diffusion, going from higher to lower concentration. higher to lower concentration. However, rather than go through just, in this case, channels as they're called in the membrane, okay, and taking their time and bouncing around, uh, like going down a water slide, possibly. These molecules zip across. They come here at a certain slow pace. They get to this opening in the membrane called the protein channel, and they zip right across. Okay, they're still going from higher to lower concentration, from higher to lower concentration. This one higher to lower concentration, just faster than normal. If you think of an airliner and you've been on a flight someplace, the most crowded place that you can think of, unless you're in first class, is going to be the cabin, the cattle car. And you are crunched in there with a lot of people. So you're in high concentration. You get out to the walkway or gangplank, depending on what they've got, and you get into the start of the terminal, and people spread out. God, they want to spread out at that point. And so you are doing a facilitated diffusion. You're going faster, and then move on and move farther apart, and then get over to the speed ramp. And if you don't know what I mean by the speed ramp, you don't even fly. I'm sorry. But the speed ramp, you get on it, and it takes you off, and that really helps the diffusion. Okay, so until that point, you have facilitated diffusion in my example. Unfortunately, it doesn't last because you'll eventually get to baggage control and passport, and everything will slow down again. Dialysis, I'm going to give 2 lines here. It'll get more when we talk about kidneys. Dialysis is the fourth of the passive processes, and it's the separation of molecules by size, just the separation by size. Old-fashioned vending machines before you were born, I'm sure. Old-fashioned vending machines, you put nickels and dimes and quarters in them instead of credit cards, and they would be separated by their size inside the machine. Okay, so dialysis. When somebody needs dialysis, they've got a lot of foreign molecules. They got a lot of waste product. They got a lot of urea and ammonia in their blood. They need to have it filtered out. The blood is taken out, and the materials are separated by size. And then, because of their size, some are excluded from reentry, and others are allowed to reenter. Okay. So dialysis works by a separation of size. Active transport. We're not in equilibrium with our environment. Okay. If we were in equilibrium with our environment, we'd be dead. Okay. Our environment around us is different than we are. We concentrate our environment. For instance, where which is where is calcium more concentrated, in your body or in the area of equal volume around you? Calcium, of course, makes up the bulk of bones. So where do you have more calcium? In you or out you?
Mm.
In, inside. Okay. And That wouldn't be the case if you were in a chalk mine, I guess, and you're lying down or covered or buried in chalk. But in real life situation, you have more calcium in you. Where's more iron? In your body, in your red blood cells, or in the space around you? In your body. How did that happen? How do you get more of something in your body than is present in the environment around you to harvest from? Because you do it over time. You take it in over time, and you take it from a lower concentration that is outside of your body. You take it from a lower concentration to a higher concentration, and that's inside your body already. So you would take, for instance, iron, possibly out of water. Okay, you would take iron and assimilate it into your body and keep it there to further increase the amount of iron in your body, which you need for your red blood cells. This is a diagram to show a membrane, and the yellow, I guess this yellow, I'm sorry, is a carrier molecule. This is outside, this is inside. Outside shows some solid. Well, we can think of it as iron if we want. Okay? And not very much of it compared to inside already. By osmosis, we would expect a higher concentration to flood out. It's not getting a chance to. Whenever one of these iron molecules gets close to the carrier protein, it's yanked in, it's pulled in. When any of these that are already in try to get back out, they're just yanked back in again. This is sort of like a prison yard. You got few prisoners on the outside and a lot concentrating inside the prison compound. Okay. Well, as you bring a prisoner new to the gate, you yank them in and crowd them in with the others. There's always the ethic that you must try to escape. And some of these prisoners are cells, or I should say, molecules, try to escape, but the guard immediately catches them back. A few probably do escape, like in real life. But so for one that escapes, 10 are recaptured. Okay? The cell doesn't care about the identity of the molecule, just the amount of the molecule. But what's the purpose of active transport? Active means with the expense of energy. The purpose of active transport is to bring enough material inside the cell to build something, to build something. If I don't have enough Legos to build a project, I got to pull them in and I got to keep them in until I've got enough. And then what could I build down here? Well, if this were an amino acid, I could build a protein. If it were an amino acid, I could build DNA. Okay. And once that DNA gets formed inside, it's too big to get back out. Active transport, very, very important. Cytosis. I believe we already mentioned this final type of process. Okay, this is where I give you the idea of a golf ball pitted and sucking in fluid and also uh, solids. When fluid is being sucked in, it's the second one. It's called pinocytosis or cell drinking. bubble of fluid is drawn into the cell. It's called a pinocytotic vesicle. When the cell draws in something that is solid, such as a bacterium or a broken piece of debris of a red blood cell, that's phagocytosis. Okay. Cytosis has two parts. Bring in solids or liquids. And we politely call them cell eating and cell drinking. Now, on the third practice test, there are questions for you on this topic. So this is why you don't rush to the practice test without reading the topic again yourself, okay? Unless you really feel comfortable with it. Okay, we've got one more set of bones to do for today's lab, and that's on the upper appendicular skeleton. And then we're going to do the lower on Tuesday, as I mentioned before, and the knee, as I mentioned. Thursday will be a complete day of review. Okay. So, any questions for you, for me? Okay, I got to do it, but there's about 4 people who have not given me their chapter assignments, 1 and 9. Maybe they sent them. I found one, my error. I found one, and I'm always looking for them, where I actually had registered the person, and I don't know why I lost track, but I got it back, and I want to get everybody. Okay, everybody is expected to do the chapter outlines. That means whether you're enrolled in uh, Bio 11, Bio 110, or both. Doesn't matter. It's the same chapter outline. You're expected to do them if you want any extra credit. Now, if you don't want to participate in the extra credit on the practicum, which is on the 9th, then you don't have to do any of these uh, outlines. You don't have to do the outlines. But I think that that'd be foolish because I think you need the outlines. I think they help. I know it keeps us all honest in reading the outlines. So I'm going to send a note and ask each of the people for whom I don't have an outline, if maybe I do have that outline, if they could send it again, possibly, or what's the situation. If I've given you a little blurb and it says, "Got it, thanks, Charlie," that's about the limit of my writing. When I get your outline, you get that, you're fine. You're fine, and that's just about everybody. One person has not taken the practice quiz. I'll send them a message too. Okay. Okay. It's 3:01. Let's take a 10-minute break to 3:11 or 12, something like that. Okay, and then we'll do the laboratory portion of the class. Okay. Okay, we're back. What we're going to do now is go over a new set of bones for the upper appendicular skeleton. The appendicular skeleton, uh, as opposed to the axial, has 126 bones. The axial, which included the brain, case, skull, in other words, and the spinal cord, that had 80 bones. This has 126. So immediately people think, oh, it's going to be harder. No, no. These are the easy ones, really. Oh, there are some hard parts, sure. But these are the easy ones because everything's bilaterally symmetrical here. And a lot of them are numbered bones. I don't know why they didn't do that all along, but I wasn't there when they did the numbering. Okay. Any questions that you have? Okay. Then I'll back on the screen. And we're down here at 128, bones and structures, upper appendicular skeleton in the lab. Last thing we have to do. You have one more thing, the practice quiz, which is, it's not up for you yet. Okay, I'll fix that. Now it's available. Okay, but please, not right now, afterwards. Okay. Uh, you want to do is your best on this bone practicum, as they call it. I think you will do your best. Most classes do. Uh, you want to get all your points that you can. You want to get all the extra credit that you can on this because you're going to be feeding off of this extra credit, these high scores. You can get 105 points. All you need is 90 for an A. So you'll have 15 points extra if you get them all. And you can whittle away at those on a test that might be harder. Um, you don't want to do poorly on an easy test. Okay. Okay, we're going into it. There we are. Okay, appendicular skeleton. That's going to be the arms and the legs later on, and the hips. It's also going to be the shoulder blades and the collarbone. Okay, that hold the arm on. We start here with an anterior view of the shoulder blade. Now, after I get through finishing these off, these photographs, okay, I'm going to use real bones and go through the same thing. I'll do that faster though, because this will prime us. Okay, you're looking at an anterior view. Anterior view means you're looking at the face of somebody like this from this angle here. Okay. This large area here is the subscapular fossa. I don't use the word body myself. Subscapular fossa. Fossa is an indentation. It looks sort of like a trowel. It looks like an ashtray. subscapular fossa. It's very nicely on your back. This point at the very top is the superior angle. A lot of us have to scratch our backs, and you find that superior angle is usually the culprit. Come straight down, and you have the inferior angle. This side here abuts the spinal column, so it's going to be medial, and this is the medial border. Medial border, inferior angle. Now go up the other side, the lateral border. The lateral border essentially is your armpit, the lateral border. It's also known as the axillary border because your armpit's also known as the axilla. Okay, but in my multiple choice questions, I'll use the ones that correspond to these diagrams, and I'll use lateral border. Okay, come back up here. Lateral angle is not necessary. You do not need to know that. Uh, if you get confused, the worst that could happen is that you learn it. It's not going to hurt. This broken little finger is the coracoid process, and behind it, the acromion. The acromion, the coracoid process, and this structure not yet named. It's not the lateral angle. It's the glenoid cavity. These attach to the head of the humerus, and by ligaments, they sort of tie it in. Coming across the top, this area is the superior border. So what looks like a triangle here has three borders: the superior, the medial, and the lateral border. An inferior and superior angle. The book is pointing out the lateral angle, but I've never seen a book do that before. Then these two structures, as we'll see again in a different drawing. The drawing below, the drawing on the left, this one, is the same as the one that we just saw. The labeling may be slightly different, but not much. And anything that is different, that's just one thing over here. Uh, we're not going to worry about it here. Okay, let's turn it around. Now you're going to pat somebody on the back. Okay, well, you're not going to find this bony structure here. It's going to feel pretty smooth because it's going to be filled below and above by muscle. The bony structure is the spine of the scapula. They're calling it scapular spine here. That's fine. It comes out. Way at the end, it flares out to the acromion, also known as the acromion process. This acromion is the same as this structure over here. This little structure here with no label, because they think it's too difficult. Not for you. This is this, the coracoid process. above the spine of the scapula. There's an indentation, a trough, a valley, a canyon, and that holds the muscle, the supraspinatus muscle. It is the supraspinatus fossa. Below it, filling this entire area, is the infraspinatus muscle, and the area is the infraspinatus fossa. Now, we've got this one in the original view, anterior. This is posterior. And what we do is just turn it so that this side faces us now. When we do that, here's the lateral border, same as this. This structure here that looks like a thumbprint. Or you want to open your phone, you press the thumbprint. This is the glenoid cavity, also called the glenoid fossa. Glenoid cavity is best. That's where the head of the humerus is going to rest against. Here's the acromion. Here's the coracoid process. And this little guy here shouldn't fool you for long. It's the spine of the scapula attached to the acromion. The spine of the scapula here attached to the acromion. This one is maddening for most face-to-face classes where I ask them to tell me if it's a right or a left. It happens to be a right clavicle or collarbone, but I won't be asking you if it's a right or a left. Don't worry. The clavicle is not properly called the collarbone. Okay. It's politely called it, but it's not the proper name. The clavicle is the most frequently broken bone in the body. Most people, if I ask of you, what bone have you broken, if any, I get the answer, the humerus. Well, that's probably true, but as people age, it is taken over by the clavicle. When people age and maybe get a little infirm or whatever, they fall, they put their hands out in front of them, and the pressure's on the clavicle. So that's really why it's the number one in breakage. I'm going to back up for a second and assume that you may not know where the clavicle even is. Huh, there's no picture of it. It comes across right here. Collarbone. Okay, if you have this bone, which is about 6 inches long in your hands, and you have one hand on one end and one hand grasping the other end, you'll notice that one end is chubby and the other is flared out, larger, but flattened. The chubbier end is called the sternal end. Why? Because it fits into the uh, sternum at the clavicular notch that we looked at the other day. Clavicular notch, sternal end. The other end out here is out at your shoulder, and it's the acromion. It's an important point of attachment for the head of the humerus. And then there's one more part, come back from the acromion, and you find the conoid tubercle here, or you find it here. You'll see the importance of the conoid tubercle a little bit later. Acromial end, conoid tubercle. Here's the body or shaft, and here's the sternal end. The humerus, okay, probably the bone known by most people. They have both the anterior and posterior view of the humerus here. Let's not worry about the view just yet. Here's the top. This fits in to this area right here at the glenoid cavity. This is the head of the humerus in both pictures. I know sometimes people think, oh, they never ask the head of the humerus. It's too easy. Yes, we do. Come below it. It looks sort of like a skull cap or a little helmet. Come below it here. This area is the anatomical neck. The anatomical neck. But down farther, right here, where you could pinch it with your fingers, and if you made your fingers into scissors, you could imagine cutting it. This is the surgical neck. This is where it normally breaks. Not at the anatomical neck, but at the surgical neck. Put your eyes on the head. The head is going to be medial here because that's where it attaches. Let's go lateral. Let's go way out. The structure here is the greater tubercle. We had tubercle yesterday on the rib, head, neck, and tubercle. Bulge. It's for a point of attachment of muscle, the greater tubercle. Now, slip back across, and we have another tubercle, the lesser tubercle. It doesn't look smaller than the greater tubercle, but that's only because the greater tubercle, most of it's on the back side. This is the lesser tubercle. We have attachment of muscle and ligament here, greater, lesser. In between, there's a groove, about the size of a pencil. This is the interturbicular sulcus, or the interturbicular groove. I don't like where the arrow's pointing there. It should be in here. Interturbicular groove. Come down the shaft of the bone to about halfway. Find the bulge on the lateral side. This is the lateral side. And that's the deltoid tuberosity. Most of us have heard of that at least. The deltoid muscle is divided into three parts, but they all attach there. And when the deltoid goes over the shoulder, okay, towards the back, it will pull the arm up at the deltoid tuberosity. Come down to the distal end of the bone. You'll notice it flares out here on the medial side, here on the lateral side. Take your hand and reach over to your other arm and find these two bumps, one on each side of the distal end of your arm. You'll find two bumps. The medial epicondyle is sharper, and the one on the other side is the lateral epicondyle. Now I've got a jacket on right now. If you don't happen to have any sleeves on, you can look at your lateral epicondyle pretty easily, especially if you have a mirror. It's that little bump. You've seen it millions of times before. Lateral epicondyle, medial epicondyle. Then down at the bottom of the bone is another articular surface in two parts. Underneath the lateral epicondyle, it's rounded here. This is the capitulum. Capitulum. And the rest of it, this part and this part, make up the trochlea. The capitulum is going to be the guide for the radius in the forearm. It's going to swing up. You'll see that better when I take out the bones. The trochlea is going to be hooked by the ulna, which you'll see better when I work it on the bones. Capitulum, trochlea. Above the trochlea, there's a cavity, and that cavity is the coronoid fossa. Part of the ulna swings up as you raise or flex your hand and fits into that. Again, you'll see it with the real bones uh, in a moment better. Above the capitulum is an unlabeled radial fossa. The radial fossa is here. That one makes more sense because the radius swings into it. I realize you don't probably have any way of writing that in right now unless you have your book open and you can write in radial fossa, or in fact, if you have your book open, you probably don't need to write it in. It's probably there. Turn the bone around. Nothing changes except there's a large cavity at the back of the bone. That's the olecranon fossa. A part of the ulna is called the olecranon or olecranon, and it fits into there. If you are able, I mean, I'm not encouraging you to, but if you're the type that can crack your elbow, make it squish. It's the synovial joint inside of the olecranon fossa that's doing that. Here's the lateral condyle over here, the medial over here. This is a little quiz when you get ready for it, but the answers are right below, and we're going to head right for the answers. Okay. Here's our head of the humerus. The star is on the lesser tubercle. The greater tubercle is over here, and the intertrabecular sulcus is here. This is the deltoid tuberosity. The radial fossa, now it's labeled, okay. I did this one, happy about that. Here's the capitulum, round. Here's the trochlea. Here's the medial epicondyle. The medial epicondyle often is known as the funny bone. We don't really have a funny bone, obviously, but there's a nerve that runs right next to it. And here's the coronoid fossa. Coronoid fossa here, radial fossa over here. Now, is this the anterior or posterior? This is the anterior. How do I know? Because down at the bottom, there's no big cavity here that we call the olecranon process. Okay, this is the ulna. A little blurry. Okay, here's the olecranon process. If you feel your elbow, feel it at the back, you feel this rounded structure. You can't miss it. This is the olecranon process. It fits into the olecranon fossa. Okay. The top of it looks like a snake, or it looks like a carnivorous plant, whichever you like. But the mouth is the trochlear notch. Trochlear notch. Now, this word, kind of blurred here, is semilunar because it used to be called semilunar notch. I won't be calling it that. I'll be calling it trochlear notch right here. And that's going to swing around the humerus as you flex your lower arm. I'm going to take my cursor, go around the trochlear notch, come down, down, and end up at this jumping off point. That's the coracoid process. If I did that over here, I'd end up right here. That's the coracoid process. Down at the bottom, this is actually the head of the ulna. It's not based upon size. It's based upon which is formed first. This is the head of the ulna, both sides. And this little bump down here, and this little bump over here is the styloid process of the ulna. Styloid process means feather-like or pen-like. Okay? And if you could have this in your hand, right now, you would see it's much like an old-fashioned pen. We come up a little higher on this one that's turned laterally. I put my thumb on the coronoid process. I can hold it here this way, but my thumb on the coronoid process. And I'm going to slip it off to the side and find this nice smooth area. This is the radial notch. The head of the radius fits into that. Okay, here's the radius itself. It's got what looks sort of like a desert feature. If you've ever been out to the desert and seen these formations. This is the head, this is the neck, this is the radial tuberosity. Do not make the mistake of calling it head, neck, tubercle. That's for the rib. This is head, neck, tuberosity. A tuberosity is bigger than a tubercle. Head, neck, tuberosity. Come down to the bottom portion. Here's the styloid process, again of the radius. The radius and ulna each have a styloid process, which is going to cradle the hand. The ulnar notch, we had the radial notch up higher, right here on the ulna. But now on the radius, we have a notch for the ulna. This is the ulnar notch. Okay, the hand. Most people think of the hand as the hardest part. I can't believe they still think that way. There's so many easy ways to learn it. Okay, I have one, and so let's get to it, and the hand won't give us any trouble. Now, what side of the hand am I looking at? The back or the front?
Posterior?
Okay, you're looking at the posterior. Thank you for correcting me. It's good. I was thinking of it when I said front. We don't use that word. Okay, this is the posterior of the hand. Okay, if you've got your hand on a mouse, for instance, right now, you're looking at the posterior of your hand. Most people are right-handed. I know not everybody, but if you're right-handed, you got your hand on a mouse, uh, then you're looking at this exact orientation. Okay. You've got 5 digits, and the digits actually start here. This, these two bones make up the thumb. We can use those terms temporarily. This is really digit 1. The thumb has 2 bones. Bend it, you'll see. A proximal phalanx and a distal phalanx. Go to digit number 2, 3, 4, and 5. They all have 3 bones. The proximal phalanx, the middle phalanx, and the distal phalanx. You need to be able to identify these precisely. It's not hard. I'll pick out this one here. Okay. This is the proximal phalanx of digit 2. Okay. How many bones did we just do? Just in the phalanges. If there's 3 in 4 digits, in all of 4 digits, and 2 in the thumb, how many do we have?
14.
Okay, thank you. There are 14 bones. We just did that. And in the other hand, we just did those. They're just mirror image. So we just took out 28 bones. 28 bones. If you weren't listening, you need to go over that so that you can identify any one of them that is picked out. Now, those are, of course, the digits of the fingers, okay, properly called the phalanges. The palm of the hand has been labeled 1 through 5 for you, okay, bones 1 through 5. These are metacarpals. Metacarpal. This is metacarpal 1 for the thumb, 2, 3, 4, and then 5 for the little finger. That's just that's how they're called. Metacarpals 1, 2, 3, 4, 5. And since there are 5 more in the other hand, we just took out 10 more bones. Now here's what gives people trouble. They don't have trouble with the long bones. They get in trouble with what looks like a jumble of bones down here. Okay, it's not a jumble. Okay, it's your wrist, and it has 8 carpals. These are called carpals. 4 of the carpals touch the metacarpals. 4 of the carpals do not. So break it down to the 4 that touch and the 4 that do not. Hang in. I think I'd like to use the lower one first. This is the other hand. No, it's not the other hand. It's the inferior side, anterior side. Okay, you'd have to turn your hand over. Okay, here's distal phalanx number 1, or distal phalanx of distal phalanx again of 1. Okay, here's the proximal phalange of digit 1. This is metacarpal 1. You can tell that it's a thumb. Come down, and it touches one of all these 8 bones. Touches essentially one. Oh, it may scrape against one of the others, but it's obviously delegated to that one. That's the trapezium. It's labeled T2 here. Okay, you can follow the labeling down below. This is the trapezium. Go to metacarpal 2. Come down, and it seems to be dedicated to this T3. We're never going to call it T3. We're going to call it what it is, and that's the trapezoid. Okay, the trapezoid, trapezium, trapezoid. Come over to metacarpal 3, come down, and it's locked onto the capitulate or capitate. And then metacarpals 4 and 5 both come in and hang on top of this one poor guy, H, the hamate, the hamate. We come back to metacarpal, trapezium and trapezoid, which are these two, and start off underneath it. And this bone is the scaphoid. It's also known as the navicular. Scaphoid or navicular. Navicular because it looked like a little tiny boat or arc, but you got to use your imagination for that one. But it's not imagination that you go from T1, a metacarpal 1, to trapezium, to scaphoid. Then come along underneath the capitate, and the next bone is the lunate. Keep going. The next bone has several names. The triangular, the triquetrum, the triquetral they're using here. I won't fool you with that because it's got so many names, I just won't ask it. But the rest are all fair game. And then their final bone is this little one that sort of looks like a pea. And that's why they call it the pisiform. Okay. I'm not cold. I just want you to be able to see the bones. Okay. Any questions? Yes. Any confusion? Of course. Okay. But you're going to get the weekend here to carving out sometime. Okay. And you're going to get to.
Charlie, I think your mic is stuck in your jacket and it's a little hard to hear you.
How's it now?
Good, that's perfect.
That was it. Hey, I've had so many things go wrong. The first day of last semester, the first time I used the microphone on the machine, damn thing, nobody could hear me. They were yelling and screaming and waving, things like that. Yeah, it wasn't working. I had bought a cheap, I guess, microphone. So I went off to Best Buy to get a better microphone. That's why I ended up with this, because Best Buy was completely sold out. Nobody had any microphones, headsets or anything. But they did have this one for, gaming headset. Okay, maybe I'll get a game later. But so that helped. Then one day I dropped it. There's a little teeny button. You probably know this. There's a little tiny button back here, and if that gets hit, out goes the sound. Okay, it'll come right back on if you know where the button is. But had to have somebody help me with that one. Anyway, we got set up, and thank you. There's one more thing that could go wrong. Okay, Oh, and also I haven't done it with you yet, but I often kick out the cable and lose it all. Okay, you're looking at me, right? Like it or not? Come on, you're looking at me?
Yes.
Okay. I said like it or not. And I want to go over the bones with you. Okay. Now, is this going to be? No, it's going to have to be higher. Oh, okay. I'm going to. Now, this is tricky because that stupid camera likes to fall. That's a bit better, huh? I'm seeing what I'm assuming you're seeing. Okay. Okay. Here's the. I'm going to go from the posterior side. Okay, of the scapula. Here's the spine of the scapula. Hang on. Here it is. Here's what I want. Be professional here. Here's the spine of the scapula. This out here is the acromion. Okay, here's the acromion. This over here is going to be the coracoid process. This is the superior angle, the inferior angle. This is the lateral border, holding it by the lateral border underneath the arm. This is the superior border. I'm sorry, medial border. It's hard to concentrate looking at yourself. Okay. The superior border is up here. Now, I'm sure I'm going to get better on the rest of them. Do you need me to do that one so far again? Okay. This is the glenoid fossa. This is the acromion. And this over here is the coracoid process. This is the supraspinatus fossa, the infraspinatus fossa, and the subscapular fossa. This is the clavicle. I said there's obviously two ends to it. I'm going to hold it by the two ends, and here we go. One end feels chubbier to me. That's that end. That's going to be the sternal end. This end is flatter, and that's going to be the acromial end. When I hold it by the acromial end and come back an inch or so, I find a bump. That was the conoid tubercle. There it is. This one happens to be a right, nope, a left, because the conoid tubercle goes down and posterior, and the acromial end is lateral, so it's going to fit here on me. You don't have to do right and left. Okay. This is the humerus. Spin it around. We're at the top. This is the head of the humerus. Well, that didn't help. Nope, that won't help from here. I was trying to show you the head, but there's not enough contrast. That's why I didn't do the axial skeleton at all. There's not enough contrast. This bump out here. Okay. This is the medial side because it attaches to the uh, scapula. This is the lateral projection. This is the greater tubercle. Here's the lesser tubercle. And I think this is a very good interturbicular sulcus. Where my thumb is not too obvious. There's a slight bump there. That's the deltoid tuberosity. It'll always be about halfway down. Nothing else could be asked of you halfway down the shaft of the humerus. Okay. This is the distal end of the humerus. This ball is the capitulum. This groove is the trochlea. Above the trochlea is the coronoid fossa, right there. The radial fossa, I have to put my finger on the capitulum. The radial fossa is right here. And it does not show well in real life either. Uh, sorry about that. Come on. Okay. That's a good shot. Capitulum, trochlea next. Above the trochlea is the coronoid fossa. Since this is the lateral side, this point is the lateral epicondyle. The sharper point is the medial epicondyle. Okay. I didn't show you the posterior side, and there's the hollow there, the intercondylar fossa. On the ulna, This is the chromium or chromium process, where I can put my finger through, let it set in there. That's the trochlear process. Put my finger back in and bring it forward to the very tip. That's the coronoid process. And then let it slide into this nice little groove here. And that's the radial fossa. At the very end, there's the styloid process. Fingers on it, up and down. Styloid process. On the other side, I won't be asking you this, but there's the ulnar notch. The radius, looking at the superior portion, here's the head, here's the neck, and here's the radial tuberosity. At the distal end, I've got my thumb on it. That's the styloid process of the radius. On the other side, is the ulnar notch. It's not showing up well. It doesn't matter because I never ask it. You know why I don't ask radial and ulnar notch is because you always show the bones together. And when you do, you fill those notches with the opposite bone. Okay. That was the radial notch.
Charlie, would you mind showing us the radius and the ulna together?
Okay, first off, we're going to see if I'm lucky. I just grabbed these out of the box. And what are the chances that I've got two right ones or two left ones? 50-50, right? So if I'm not, I'll probably beg off until Tuesday. And yes, I can do it. Let me work with it down here out of sight. Okay, that fits. I did get lucky. Okay, just looking at the head portion first. See how they fit? Okay. You see the acromion, you see trochlear notch, come straight out to the coronoid process. The head of the radius, okay, fits into the radial notch when I pull it apart. Don't make me hold it here all day. Tell me it looks okay.
That looks good.
I think it does too. Okay, we don't need to worry about the center portion. Mm. Okay, I got them as close as I could manage here. Okay, This is styloid process of the radius, and this is the styloid process of the ulna.
Thank you.
Where,
That
okay, that's good. Now, last thing to do is give a try on the bones of the hand. I think we'll be okay because this is kind of stretched, and that's good. We need that. Okay, on the thumb. There we go. Here's the distal phalange. Here's the proximal phalanx. Sorry, didn't realize I was dropping it. Distal, proximal, metacarpal 1. Let's just move over to metacarpal 2, the middle one, metacarpal 3, metacarpal 4, and here's metacarpal 5. There we go. That's a little bit better for me. Okay, here's uh, okay, metacarpal 1 is what I'm holding. Holding the thumb. And below it, this carpal that I'm touching, this is the trapezium. Metacarpal 2 here, and below it, right here, that's the trapezoid. Okay, trapezoid's at the tip of this fingernail, and this one. Go over to number 3, metacarpal 3, here. Come down to this one. This is the capitate. Go over to metacarpal 4 and 5. Hang on there. These are 4 and 5. And below it is this one, the hamate. And it does sort of look like a ham hock. You can see that it's got a bony projection. Okay. As far as showing the other 4, I know you guys are just being sadistic. You know, it, it's not going to come out well. So I don't think it'll come out as well as photographs that I put up and other photographs that I can put up for you. Uh, uh, I always had the feeling when I was in school, if the teacher can't do it, I shouldn't have to do it. Okay, but you do, and you can do it from photographs. You can also do it from YouTubes. I'll post a couple YouTubes that were done in private by another instructor of ours. Okay, and now what does that symbol mean? Anyway, Stephanie, did you need something? Okay. Uh, uh, don't get dizzy on me or whatever. I'm going to move this camera. I'm going to try it on the tabletop. Holding it up like that's terrible. I'll close the camera first. Okay, here are the bones. This side is the thumb side. This was the trapezium. Coming down to the second row, this is the scaphoid. Going under that, this one is the lunate. Why do they call it the lunate? Because it looks like a moon. Not to you. You got to turn it sideways. And it does look like a moon. The next one over, this one here, is the triangular or triquetral. And this one usually sticks up. This one is the pisiform. Going back to these, here's metatarsals 4 and 5. Excuse me. 4 and 5. This is the hamate, metatarsal 3, the capitate, metatarsal 2, trapezium, metatarsal 1, trapezoid. Going around again, scaphoid, lunate, triangular, and pisiform. Okay, now I feel better. Any final questions you guys got? How do you feel about the test coming up?
I feel pretty good about it as long as I just study for it and memorize parts and everything. Then I feel like I'll do good.
Okay.
Same here.
I
Yeah,
had
same.
it, reading the discussions. Actually, I read from last year, and I was reading what people thought. And this one, oh, I always say girl. I'm not supposed to. I'm not allowed to say it. What are they going to do to me? I can't believe it. I'm supposed to say woman or female. You want, what do you want? Woman or female? You can have whatever you want. I mean, I asked you to call me this and that, but I mean, they really can't do much to me. Uh, but uh, You need a way to sometimes to describe somebody, not belittle them or anything, but just to describe somebody. I can't use the term ma'am. You know, I'm not allowed to use the term ma'am because that's derogatory. It's a reference to age. Okay, somebody said that anyway, and I don't know. But the guys, I can call them bro, dude, bastard. I can call them anything I want to. I'm part of the group, you know, and people are people. So if I offend you, tell me and I'll stop somehow immediately. But I'm not trying to, obviously. But anyway, I was reading this one submission, and the girl was saying, I was overwhelmed by the memorization until I figured out how to do it, and then it was a piece of cake. So you just uh, try different ways if you can. Uh, uh, I like straight memorization, but other people have different techniques. I'll ask that as a question on discussion if I haven't already, for you guys to talk to amongst yourselves. What do you find is a good method of memorizing? We all do it. How do we remember our vocabulary, our parents' names, our friends' names? We memorize all our life. It's just comes down to me. Do you want to memorize it? Okay. Well, think of that A. Think of the A at the end. Okay. So email me with any concerns that you have, and I'll let you go. I will stay until the last person's gone, in case anybody does have a question. They don't want everybody here. Okay, but you're uh, welcome to exit and have a good evening.
Thank you,
Thank
Charlie.
you.
Bye, Charlie.
Charlie.
Thank you,
Bye.
Charlie.
Bye. Thank you.
Bye, thank you.
Thank you.
Thank you.
Bye, Charlie.
Thank you.
You have a good night.
Charlie, when did you say the test was again?
Okay, now you got to be able to find your syllabus, but it's the 9th.
Okay, perfect.
Okay.
Thank you.
Haley, are you still there?